Hello, my name is Leslie Muniz, and this is an identification of uncommon antibiotic producers in Illinois soil isolates. Many people believe that our bathroom is the dirtiest place in our home, but it's actually the kitchen because of the foods and the decaying food and that bring more germs. Also with that, many people believe that hospitals are the cleanest place that anybody could be in, but because of the germs from other patients, many other patients that are not there for these infections can acquire infections, which are called nosocomial bacteria, from these nosocomial bacteria. But as we know, bacteria form a big part of our lives, good or bad. Bacteria are in soil, and also we are heavily involved with soil, directly or indirectly, and because of the resistance in bacteria, there's a crisis of antibiotics, and also with soil because of other contaminants. And the TEPI, which is the Tiny Earth Project Initiative, they strive to educate students about these crises and also give them the tools for original research to find new antibiotics against specifically escape strains that are multi-drug resistant and virulent. In this figure, I obtained my soil from under these decaying leaves, and it was a fairly rich soil. And what we wanted to use them for is to test the isolates obtained against safer strains such as Bacillus subtilis and Escherichia coli. And what we wanted to use them for is to identify the isolates in the soil if they are common antibiotics such as Bacillus or Streptomyces. What we basically did is use the soil samples and create serial dilutions because of how concentrated bacteria may be in the soil. We wanted to dilute them, and we used the last few dilutions that we had, and we put them on spread plates, 3 different media, analyzed the growth, and basically just try to find antibiotics, reconfirm them, calculated antibiotic producers, and also what we could have done with the individual colonies from our streak plates is we could have run a PCR, but it was unsuccessful, so we could not really extract DNA and analyze the 16S RNA sequences from using BLAST. So what we could do is conduct a gram stain, a series of biochemical tests, and also use colony morphology. In our first image, it is an image of our confirmation of antibiotics. So both unknowns, but unknown A was ID number 19, but it was on a TSA media. And unknown B was ID number 19, but it was originally on a PDA media. And as you can see, they did form a clear zone of inhibition. Unknown B was had a broad spectrum, so it had tested against E. coli and Bacillus. And then from our biochemical results, which could have some discrepancies because of unknown variables that are that we are not specifically sure about, that we'll see later on the table in our next figure. But the biochemical tests that we performed, and specifically in the tubes, were for fermentation of lactose and glucose, and then the last tubes were for triple sugar iron. And these are more differential tests compared to the plates, which are more differential and selective, as we will see in the table. So first off, analyzing this table, McConkey's was actually negative for unknown B, and red signified that it fermented lactose, but as we see in the phenyl red with lactose, it was negative, so there's contradicting results from that. And yes, unknown A and unknown B had very contrasting results compared to each other, just like our tester strains. And the results for Bacillus and Streptomyces, which we didn't test but we found in literature, was very different compared to the unknown A and unknown B, and did not really concur with each other. The most common phyla of soil bacteria are proteobacteria, yet most antibiotic producers were Firmicutes and Acinetobacteria. Because of results that we obtained, we could not really confidently identify unknown A or B, but as we see with B, it might be a stronger antibiotic, maybe even a pathogenic bacteria. But we would need to confirm these identities would be 16S rRNA isolation of the DNA and use blasts to identify the genus. And then afterwards, we could purify the samples and obtain the proper chemical agent that's being produced from it, and hope to understand the biochemical pathways and hopefully to recreate synthetically made antibiotics against these escape strains. So, yes, I appreciate your time, and hopefully this is helpful for your research. Have a good day.